give this talk, I actually asked for permission to extend it just a little bit. So I'm going to put a little asterisk there. Oop. I'm going to add DLL3. I think partly because there was such exciting data presented at World Lung this year on small cell lung cancer with an agent against DLL3, and also because it gives us a more vivid view of how Notch as a target may be very interesting. And even in this era of kind of sexy immunotherapy and targeted therapy, stem cell approaches still have some currency. My disclosures. So again, the Notch pathway mediates intercellular signaling and stem cell self-renewal, proliferation, and differentiation. Receptors include Notch 1, 2, 3, and 4, and ligands, a delta-like ligand 1, 3, and 4, as well as jagged 1 and 2. DLL4 is a particularly compelling target because it's required for vascular and immune development, and it's upregulated in tumor angiogenesis. So there's a hope that by you are not just inhibiting Notch, but also having some anti-angiogenic effect as part of the mechanism. DLL3, which I'll return to, doesn't seem to have as much identified role in tumor genesis, but there is in embryology importance in the development of the spinal column. So demsizumab was covered yesterday during the showcase, and for those of you who didn't see it, I'm just going to quickly review. But demsizumab is the first-in-class antibody against DLL4. It's a humanized antibody, and we'll talk about the mechanisms of action in more depth on the next slide, and the half-life is approximately 16 days. Again, the mechanism of action is actually threefold, at least it's purported to be. There's cancer stem cell approach, and the idea just by inhibiting Notch that there's differentiation of the cancer stem cell and then chemosensitization of those cells upon differentiation. But again, choosing DLL4 as a target in particular is also intended to block the critical role that DLL4 has in tumor angiogenesis. And then finally, I'll review some of the data that Oncomed showed at AACR this year that suggests there also may be some role in stoking the immune response to the tumor. So to briefly review the first-in-human study, this was a standard dose escalation phase 1 first-in-human, and the take-home was not so much the DLTs that were noted, and a lot of the side effects had to do with expected on-target effects like hypertension. It was a late emergence of congestive heart failure in 4 of the patients on study, and this was after a significant cumulative dose and was fairly serious in those patients. It turned out that going back to the drawing board upon finding this, that developing a risk mitigation strategy that includes very frequent BMP testing and echocardiography parameters for discontinuation, that's actually been mitigated quite nicely. Here's the waterfall, which is certainly respectable for a first-in-human study in a heavily pretreated population, stable disease plus partial response rate of 40%. And here's the swimmer plot. I'll take an aside. This was my very first phase 1 study as a fellow, and I had 3 of the 4 sick CHF patients, but I also had that gold medal swimmer on the bottom too. So it just goes to show the joy and agony of developmental therapeutics. But I was happy when they did go back to the cardiologist and design a risk mitigation strategy and incorporate that into the phase 1b that was dedicated to non-small cell lung cancer with carboplatin and pemetrexed. The study design had several cohorts, and I'll summarize by saying that there were a couple of changes that happened over the course of the study. First, as they developed their risk mitigation strategy, they went from using maintenance demsizumab to just a truncated dosing schedule with just 4 cycles alone, with those 4 cycles of chemotherapy. In addition, there was a secular change toward pemetrexed maintenance toward the end of the study, and so the ending cohorts 5 through 7 all followed that standard of care with ongoing pemetrexed maintenance. Notably, as I mentioned, with institution of these changes, there was not a single case of congestive heart failure noted on study. There was hypertension that was easily managed. There was BNP that was found that was asymptomatic, all of which resolved upon discontinuation of drug. And here's the waterfall plot, which is actually pretty remarkable. This is certainly respectable for a carboplatin pemetrexed regimen with a CR plus PR rate of about 50% and a clinical benefit ratio approaching 90%. And there didn't seem to be any decrement of effect in the truncated patients in whom only 4 doses of the demsizumab was given. Here's the overall survival, which is not yet mature. Still haven't hit the median overall survival. But these results were good enough for Oncomed to invest in their phase 2 trial called Denali. This is a carbo-PEM plus or minus gemcitabine in non-squamous, non-small cell lung cancer. They're taking 200 patients, and it's going to be a 1-to-1 randomization with carbo-PEM for 4 cycles, followed by PEM maintenance, and half the patients getting gemcitabine for those first 4 cycles. Overall survival is going to be the endpoint for this study. So at the showcase, Leo very briefly alluded to this, but I wanted to present a couple of the experiments that they presented at AACR this year. Basically, they took a murine colon cancer model and added DLL4 antibody, and among the findings were that it decreased IL-17A and increased the splenic sequestration of the MDSCs. So that was DLL4 alone effect. But then they also looked at DLL4 plus anti-PD-1. And what they found was a variety of changes that kind of speak towards some synergy, particularly that it was more interferon 2, more interferon gamma, IL-2 and interferon gamma suggesting a type 1 response. But for a clinician like me, this was the most compelling slide, which is just the tumor volume. The pink line is the control mice, the yellow is PD-1 alone, the blue is gemcitabine alone, and the combination seemed to have a synergistic effect in terms of tumor volume. And so this was compelling enough, and of course immunotherapy is sexy enough, that there's now a phase 1b study planned of gemcitabine plus pembrolizumab in solid tumors. Also of note, they're actually thinking about, if assuming the safety looks good on this, to have an expansion cohort of first-line non-small cell lung cancer patients who will get carboplatin, pemetrexed, pembro, and dem. So a quadruplet therapy, carbo, pem, dem. So that'll be a mouthful, but it'll be interesting to see. There are other DLL4 antibodies that are being developed. Regeneron has anotikumab, which reported out phase 1 results this year in CCR. They had a modest response rate. They had toxicities very similar to gemcitabine, again, notably hypertension. MedImmune also has a drug. They reported immature phase 1 results at ASCO as well this year. The main difference that I can tell between these two and the gemcitabine compound is these are human antibodies versus DEM, which is a humanized antibody. Also of note, in Oncomed's portfolio, they've also developed a bispecific antibody, which has activity both against DLL4 and VEGF. That's currently in phase 1. They haven't reported out any results, but it'll be kind of interesting to see if that improves outcomes. So moving to a slightly different target, over to DLL3. Again, DLL3 is another one of the ligands in the Notch pathway. And what was noted by the company developing this drug is that it's aberrantly expressed in a small cell lung cancer tumor infiltrating cells. And that prompted some investigation in that field. Here's the actual drug. It's called Rova-T, rovalpituzumab tesirine. The antibody against DLL3 is yoked to the drug toxin, and Stemcentrx is the sponsor. In DLL3 by IHC, you see that actually a large proportion of small cell lung cancer patients have high expression of DLL3, depending on the mechanism, 60 or 75%. The study was a phase 1 dose escalation, and as they climbed the dose levels, they found the DLT to be serosal effusion and thrombocytopenia, so backing off to the doses they used in the expansion cohorts. Fortunately, they still found some confirmed responses, so there was a good therapeutic index there. The AEs in the expansion cohorts were very modest, notably the serosal effusions and the thrombocytopenia, or there were no serosal effusions, grade 3 or 4, and modest thrombocytopenia at the dose. And here's the waterfall plot. So for a relapsed refractory small cell lung population, that's a pretty respectable waterfall plot in itself with an overall response rate of 20%. But I'll draw your attention notably to how much green there is to the right of the waterfall. Those are the high expressors of DLL3. And here's those patients broken out. And even a more impressive waterfall, a second line response rate of 40%, and a third line, getting to John's question earlier, this is a third line response rate of 38%, which is not so bad. And here was a swimmer plot, which also showed some prolonged responses as well. So I think it's very provocative, obviously early stage, but I think when the small cell lung cancer gets the short end of the stick, data like this certainly make us excited about what's to come. Again, just speaking to certainly superior to topotecan and pointing to the idea that it's, to think that there may be a biomarker-driven strategy in the further development of this drug, it makes sense. So in sum, drugging Notch in lung cancer is certainly a viable approach. Anti-DLL4 has been shown to be a potent strategy. There are at least potentially 3 mechanisms of action against cancer stem cells, against angiogenesis, and against tumor immunity. Demsizumab is the furthest along in trials with an active phase 2 program in lung, and I didn't talk about pancreas or ovarian. And stay tuned for kind of some provocative studies as an immunotherapy combo with anti-PD-1 agents. DLL3, promising early data in small cell. Look forward to hopefully Stemcentrx funding of the next phase of trial. And finally, I'll make a pitch for maybe next year, maybe having 20 minutes for these talks. I think there's other stem cell pathways of interest. Notch2 and 3 antibodies are on the market, going beyond yesterday's gamma secretase inhibitors. And certainly other pathways like Wnt, RSPO, and Hippo, I think, are coming to the fore. So I think these are a really important set of pathways that are worthy of further exploration. Thank you very much.
Very nicely done. So the DLL4 antibody cardiotoxicity, what's the mechanism and is it reversible?
So when the risk mitigation, so I don't know the pathway I think is non-target. We know that this is involved in angiogenesis. That's the best I can do, waving my hands a bit. What I'd say is that since the institution of the risk mitigation strategy, all the patients were found before clinical heart failure with elevated BNP or a decrease in EF, and all those patients had reversibility of those findings. I can't speak to the initial patients who were found on study.
I think your point about stem cell targeted therapy being an untapped, or I don't know if it's untapped, but I think directing therapeutics there is a great idea. My question is though that most of the mutations in solid tumor for Notch are loss of function, and if you look at the mutations of genes that are sort of feeding into the Notch pathway and TP63 activation, the Ajuba mutations that I mentioned a little bit briefly, FAT1, all seem to sort of be inactivating Notch. So how does that fit into all of this?
Excellent question. I don't think I can speak intelligently to that. Your colleague behind you might. It's a fair question, though, and I don't know that we've been able to find any of that in this patient population. They haven't, I think, investigated Notch particularly that way.
Yeah, this is Jacob Dupont from OncoMed and work with Dr. Gubens. But just regarding this question about the mechanism of toxicity from blocking of DLL4, it's actually some interesting biology, because when you block DLL4, you actually cause a commensurate rise in VEGF, or vascular endothelial growth factor, which actually recruits endothelial cells either into the space of the tumor or into certain organs like lung or cardiac. But the net effect is it's actually a non-functional vascular supply, and the net effect is actually decreased perfusion. So that's what happens. And when you stop blocking DLL4, you get rid of that VEGF spike, and the endothelial cells essentially dissipate, and thus it is a reversible phenomenon when you have that decreased perfusion in places like the heart and the lung. And certainly that is why we moved into this risk mitigation plan of just giving therapy for a defined period of time of approximately 70 days. And then you actually don't get to the point that you have so much recruitment of those endothelial cells that are non-functional that actually cause a compromise of those normal organs. But it's actually reversible, and that's something that we've proven in the cynomolgus monkey tox studies as well through the recovery phase, where you can see resolution of these lesions.
Thanks.